Hello, everyone, and welcome to today's MetroFlow event. It is 1 o'clock, so I'm going to give another minute or two for more people to join in. If you want to, for some Zoom housekeeping, if you want to select view option, fit to screen or fit to window, we found that's the best viewing model. And we'll be using the Q&A button at the bottom of your screen for questions during this time. So to get started, it's been sort of fun to see where people are joining us from. So if you want to use the Q&A, let us know where you're watching from. Thomas in New York City. Ooh, Yardley, Pennsylvania, Long Island, and Montreal, Quebec. Thank you for joining us. Okay, I'm going to go ahead and get started. Hello, everyone, and welcome to the third event in our MetroFlow Corporate Member Launch Seminar Series. I'm Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we are not able to meet in person during this time, the 15 members of the MetroFlow Steering Committee have created this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience and beyond via this virtual platform. This session is the third of several to follow through the remainder of the summer, and you can find our previous webinars on the metroflow.org webpage. As a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have the opportunity to submit a proposal to present in this launch seminar series. And the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. Please use the Q&A button to submit questions during the presentation and use the chat function to report any technical difficulties. Before we get started, we'd like to thank today's speaker, as well as you, our MetroFlow members and extended audience, for your time and participation. And with that, I'd like to introduce our speaker, David Amir of Astrolabe.
Thank you, Tina. And I want to thank MetroFlow for allowing today's presentation to happen, especially Tina and Kathy. And also, I'd like to thank the audience. I know that labs are slowly going back to work, so everyone is getting busy again. I appreciate the fact that you took some time out of your day to listen to today's presentation, whether you are live here with the audience or whether you're watching a recording. And today, I'm going to talk about one of the new modules that we released for Astrolabe for detecting differentially expressed biomarkers in complex flow and mass cytometry data. Just a brief reminder, if you have any questions during the talk, please use the Q&A button below. Just click it, enter your question, and I'm going to answer it during the presentation. Briefly about myself and briefly about Astrolabe, I'm a computational biologist. I originally come from the CyTOF. Notably, I'm the first author on Disney, so if you're tired of these dots, now you know whom to blame for them. I founded Astrolabe 3 years ago, and at this point, we analyzed more than 1,400 experiments from 50 different institutions using the platform. And the motivation for Astrolabe is this idea that as flow projects become more complex, their analysis is exponentially more difficult. And by difficult, I'm referring to time, money, biological expertise, computational power, any of the different resources that you would use when analyzing your data. And I think this audience is intimately familiar with this area of the curve where you're dealing with traditional cytometry, you have 8 or 10 or 12 markers, and you can use traditional gating or straightforward statistics to analyze your data. We now know from the CyTOF, the Aurora, the Beta Symphony, that as your panels become more complex, analyzing them is much more challenging. Gating is unfeasible because of time constraints and because of high variability as you add more gates. And furthermore, there's many statistical pitfalls, which you would like to address as early in the project as possible. Thankfully, there's a wide variety of tools out there that you can use for analyzing your data. There's many different software platforms. Some are online, some you can download to your computer. Plus, there's different programming languages like R and Python. And I like to envision these as the Swiss Army knife of flow and mass cytometry analysis. These software packages are excellent if you know what you want to do and you know how to do it. And just to name a few of the tools that they have available on the bioinformatics front, they have t-SNE, they have clustering, they have SPADE, and of course the R programming language has many different packages. However, from conversations with researchers, both bench scientists and bioinformaticians, the Swiss Army knife can sometimes look like this. There's a lot of different algorithms you can choose for. There's different parameters you need to set, and it's very easy to get lost with the wealth of information, especially when dealing with a new data set. Astrolabe is designed as a staging ground for your analysis. And the idea is that we offer a standardized pipeline for the analysis of flow and mass cytometry data. You acquire your data, whether it's human or mouse or non-human primates, and your instrument of choice could be the CyTOF, could be the Aurora, could be the Fortessa, really any kind of flow cytometer. And then this single cell data goes on Astrolabe, and the platform takes care of all steps of the analysis pipeline. So Astrolabe can clean the data, can identify different cell subsets and conduct unsupervised clustering, and finally fulfill the role of the biostatistician by identifying biomarkers that differ across experimental conditions. This entire series of algorithms and tools are fully automated. There is no gating required on your side. You don't need to choose any algorithms. You don't need to set any parameters. Astrolabe automatically takes care of these different steps. And then within 1 to 2 hours, you can log in and use our intuitive interface to navigate through the results. Before I start talking about the new module, I know that for some people, Astrolabe is a new concept. So I'm going to demonstrate the platform using a familiar data set. And in this case, I'm using an immune monitoring data set that was acquired by Professor Adib Rahman in Mount Sinai. In this case, we're looking at chikungunya infection. And this is part of a larger study that was done by the Human Immune Project Consortium. In this study, we have 42 patients. For each patient, we have 2 different time points. And these samples were acquired using many different techniques, including RNA sequencing, including 40-plex Luminex, and specifically for the purpose of this demonstration, using a CyTOF, a 33-marker CyTOF experiment. The goal of this study is to compare the structure of the immune system between these 2 conditions, the acute and the convalescent. So I'm going to switch to Astrolabe, and I'm going to show you how this data looks like through the lens of the platform. Now, as I mentioned, our emphasis is to be your staging ground. We want to get you from raw data into meaningful biological insights as efficiently as possible. And what you're seeing here is everything that you as the researcher need to do to work with the platform. First of all, you upload your FCS files, and Astrolabe is cloud-based. Everything is hosted by Amazon. Then you set up your experiment design. In this case, we have 42 patients, and for each patient, we have 2 different time points, acute and convalescent. This table is like a spreadsheet. So if your experiment has different treatments, different mouse models, different outcomes, and so on, you can encode all of these here, and they will be used as part of the analysis. So you do this, it takes about 10 minutes. And that's it, you're done. Astrolabe kicks in, takes care of the analysis, and within a couple of hours, you can come back and navigate through the results. I mentioned that our emphasis is on biologically meaningful insights, and I'm going to show you a few of the tools that Astrolabe offers in this context. I'm going to start by switching to the cell subset navigator. This is a composite view of the entire experiment. So everything I'm showing you here is using data from all of your FCS files, whether you have 5 or 50 or 500. And the starting point are all of these different cell subsets that Astrolabe identified automatically within your data. This is done using an automated algorithm. And specifically, the underlying strategy is very similar to traditional gating. Notably, for example, we start by identifying T cells based on CD3 and lineage negative. Then we look at CD4 versus CD8. And then we look at different markers that correspond to naive, memory, and Tregs. This system is flexible. If you use different markers, we're going to use them for identifying subsets. Furthermore, it is customizable. So if your panel has specific subsets which Astrolabe does not detect, we can work with you on customizing these rules. This is part of the basic service that we offer. So going back here, we have the different cell subsets that Astrolabe identified. And then we have a visualization called the MDS map. In this view, each one of these bubbles is one of the cell subsets, and their position corresponds to the similarities between them. So if I color code this by CD3, we'll see that all the T cells are next to each other. And if I color code this by CD20, then here are the different B cells. Myeloids are up here, and here are NK cells. So from the get-go, you can communicate with the data using the language of immunology. You can talk about B cells and naive T cells without having to worry about annotating T-SNE maps or about labeling clusters. And finally, the dimensionality reduction view allows you to organize these different subsets in the high-dimensional space. One of the analyses that Astrolabe offers, and we've been offering it since launch, is called differential abundance analysis. Differential abundance analysis is all about frequencies. We would like to compare whether a given population is more or less abundant across experimental conditions. And in this case, we can activate the differential abundance analysis for the time point feature. And when we do, the different cell subsets will be color-coded based on the fold change between the acute and convalescent time points. Just by looking at this, we can see that one of these bubbles is bright red. If we click on it and scroll down, we can get additional information just about this subset. So this section focuses on the intermediate monocytes. And the first thing we see is that we have aggregate statistics across all of the samples. The blue box is the acute time point. The orange box is the convalescent. The white line is the median. And we can see how it starts at 3% and then drops by sixfold to 0.5%. Then we can see the same information on a sample by sample basis, in this case, patient by patient. Every pair of bars is one patient, and we can see how the same trend persists across all of the patients in this study, with the exception of a handful where the trend is actually reversed, and the convalescent time point has more of these intermediate monocytes than the acute time point. And finally, this is all quantified using a statistical test. The FDR score you see here is equivalent to the p-value. The higher it is, the more significant the results. So you can use these figures, you can use these numbers, you can plug them in your next grant application because everything is backed by the statistical test. To recap, we're using the panel to identify the different cell subsets. We're using the experiment design to give you this kind of intelligence. And the goal is really to get you from the raw data to these visualizations in just a couple of hours. Notably, these cells were featured in the paper that came from this study. Back in the day, it took about 20 weeks to analyze this data set. With Astrolabe, we can reproduce these results in just 5 mouse clicks. So this was a brief introduction to Astrolabe. And again, the emphasis is on getting you to meaningful biological insights. Now, let's talk about some of the new stuff that we released over the past month or so. And I'm referring to a completely new module that is called Differential Expression Analysis. Now, everything I've shown you so far is talking about populations, naive CD8 T cells, or memory B cells, or intermediate monocytes. And the comparison I've shown you was for population frequencies. In other words, how does the quantity of these different cells varies across experimental conditions? However, a very common question in flow cytometry is how do specific markers change in their intensity across conditions? In other words, we're looking at different samples, let's say responders versus non-responders, and we have different cell subsets, T cells, B cells, and so on. And we would like to add a third dimension, comparing the different markers. A very common question in this context comes from immunotherapy. How does the level of different exhaustion markers, such as PD-1, CTLA-4, etc., change in different T cell populations across responders and non-responders? This is also very similar to using MFIs in your analysis. or trying to put a gate on a marker like a PD-1 on the long tail to quantify positives and negatives. So one of the tricky things with this kind of framework is that we have three different dimensions here. The x-axis is the different samples, the y-axis is the cell subsets, and then we have a third dimension for the markers. It's not really comfortable to think about. So what we're going to do is we're going to collapse two of these into just one axis. So what we're trying to compare is pairs of populations and markers that change in expression. So how does PD-1 in the T cells change? How does LEG3 in the T cells change? And so on and so forth. So I'm going to demonstrate the new module with Astrolabe, and I hope I can illustrate how it can bring you to these kinds of insights and visualizations very effectively. To do this, I'm going to use another study, again from Mount Sinai, and this one is toward the CS et al. And this study is about the immune monitoring of peanut allergic patients. Specifically, they had 6 patients who were allergic to peanuts, and they had 3 healthy donors. They took the samples from these 9 individuals, and they exposed them to 3 different conditions. The first one is media, which served as negative controls for activation. Then there's anti-IgE, which serves as positive control for activation. And then finally, there's the experimental condition, which is the peanut extract condition itself. Each one of these have 2 time points, 15 minutes and 30 minutes. And when you sum up the combinations, you end up with 54 different samples that we would like to compare in this experiment. Plus, we have a 30 marker panel. About 10 of these should be responsive to activation. So we have quite a lot of hypotheses that we would like to test in this context. So let's switch to this experiment in Astrolabe. And this is a bit more complex than the previous experiment. Before, we just looked at 2 different time points across patients. Here, we're seeing the experimental design as it was encoded within the platform. So we have the patient IDs, and then we have controls, and we have patients down here. We have the different time points, and we have the different treatments. And finally, we have these auxiliary features. And the purpose of these features is to allow us to ask very directed questions of our data. For example, the patient 15-minute column includes only samples from the patient samples and the 15-minute time point. There's 3 different conditions: media, anti-IgE, and peanut extract. And this column allows us to focus just on this aspect of the experiment. Similarly, patient 30 minutes is the 30-minute time point for patients, and then control 15 minutes and control 30 minutes are these respective time points for the control donors. Once we have this in place, we can repeat the same workflow that we've seen with the chikungunya experiment. So I'm going to go to the cell subset navigator, and we have the different cell subsets that Astrolabe identified. Again, this is a pretty rich data set. And we can see how the abundances of different populations changed in response to these experimental conditions. So let's look at the patients, and let's compare the 15-minute time points. And if we look at the MDS map as before, there really aren't that many differences. In fact, we have a second visualization called the volcano plot. In the volcano plot, every dot is one of these subsets. And those dots that are above 1.5 are statistically significant. In this case, there is nothing statistically significant. In fact, if we take a step back and just look at the B cells, the myeloids, the NK cells, and the T cells, and then compare them across the different experimental conditions, there really isn't that much of a difference. So let's sort these by patient ID. And now every 3 bars is 1 patient, and we're seeing the anti-IgE, the media, and the peanut extract. And we see that the frequencies for a given patient are more or less the same. Honestly, this is what we would expect. We don't expect cell populations to change dramatically within a 15 to 30-minute period of exposure to a given stimulation. However, we do expect these cells to respond. So I'm going to go to the new analysis tab. And when I look here, we see the different analyses that we defined before: the control 15 minutes, the patient 15 minutes, and the 30 minutes for these as well. So I'm going to start by looking at the 15-minute time point for the patients. And what we're seeing here is a heat map where each one of the rows is one of the cell subsets, and each one of the columns is one of the markers that we wanted to analyze. As before, we have different colors. The deeper the color, the greater the fold change for that marker subset combination. And just by looking at this, we can see that there is several responses. For example, it looks like plasmablasts respond a bit based on ADR. However, the cell subset that responds the most by far is the basophils. And just full disclosure, this paper was designed to measure the basophils specifically. So it does validate the study, and it does help us feel encouraged that this is the population for which we see the most prominent response. So I'm going to click on the basophils, and I'm going to choose two of these surface markers that seem to increase in response to the stimulations. So we're looking at CD61 now, and I'm going to focus on the feature box plots for this marker. So what we're seeing in this box is just one cell subset, the basophils, and we can see the level of CD61 in each one of the samples. Each dot is one sample. Here are the media ones. Here's the peanut extract, and here is the anti-IgE. And we can see how the level of CD61 increases between the media and the peanut extract. In fact, it goes as high as the positive control. I can scroll down and look at CD63. There seems to be a bit more variability here across the 6 different individuals, but we still see in aggregate the same response. The level of CD63 starts here on the baseline for the media, and then it goes up for the peanut extract and for the anti-IgE. If we would like to learn more about each patient in isolation, we can click on the sample dot plots. And now each one of these columns is one of the patients, and we can again compare the 3 different conditions. I'm going to make sure this is sorted by patients. So every 3 dots here is one of the patients, and we can see how 4 of them, 1, 2, 3, and 4, respond quite well, at least based on CD63. 2 of them, these 2, don't seem to elicit a response in neither the peanut extract or the anti-IgE. Then going up to the CD61, we can see the same trend. Now, let's compare this to the controls. So I'm going to go back to all of the analyses. I'm going to go to the controls. And once again, I'm going to choose the basophils and look at CD61 and CD63. And then I'm going to scroll down and look at the feature box plots. And we see that the media and the peanut extract are the same. So in this case, the peanut extract condition looks like the negative control. And we can see that the positive control is responding quite well over here. And looking at CD63, again, a similar trend. So on one hand, we have the patients, and we see that they respond pretty well, at least based on CD61 and CD63. Then we have the controls where the peanut extract looks about the same as the media. Now, everything we saw here so far was an aggregate. Can we examine these trends in a single cell resolution as well? And Astrolabe easily allows us to do it. So I'm going to go to the summary page, which shows us all of the different samples. I'm going to start by clicking on the media sample. And I want to examine the basophils. And specifically, I would like to look at CD61 versus CD63. And within a few seconds, I am going to have a biaxial plot that shows us the level of these 2 markers in the media conditions. Then I'm going to go back up, and I'm going to look at the peanut extract, again, for this same patient. I'm going to generate the same plot. So basophils, and I'm going to look at CD61 versus CD63. And scrolling down, looking at the biaxial plot, we're going to see that there's much more of a response up here. So let's go back to the different analyses. And so far, we looked at 2 different markers, CD61 and CD63. Furthermore, we compared them just for the patients and the controls separately from each other. For the next analysis, I'm actually going to show both the sample type, whether it came from a patient or from a healthy donor, and the treatment. So we can do a cross-analysis that combines all of this different information. So I'm going to click on this type X treatment for 15 minutes. And once again, we're looking at this heat map, and this time we're seeing much more responses. It's important to point out that we're seeing these because we're also comparing the controls to the patients, and there might be some inter-individual variability there. Just to validate the results we've seen before, I'm going to go to the basophils, and I'm going to click on CD61 and CD63. And I'm going to show the feature box plots. So we're seeing the three control conditions: media, peanut extract, and anti-Ig. And we're seeing the three patient conditions: again, media, peanut extract, and anti-Ig. And we can see that in both cases, we're seeing a response for the anti-Ig condition, but we're only seeing a response for the peanut extract within the patients, not within the controls. So same data, just a different way to look at it. Some researchers prefer one visualization compared to another. This data also has three phosphorylation markers for P38, ERK, and S6. So let's transition our attention to these three markers and see how they change across the basophils and the different experimental conditions. So scrolling down, we're looking at P38, and you'll notice that this visualization reproduces what we've seen with CD61 pretty nicely. Once again, the controls and the medias look about the same, and then peanut extract, we only see a response for the patients. Looking at ERK, interestingly enough, there is a bit of a difference here. You could argue that there is a bit of a response for the peanut extract, for the controls when it comes to ERK. Now, it's difficult to know whether this is real or not. Thankfully, from a reporting perspective, one of the things that Astrolabe offers is a spreadsheet with all of the statistics for this data. So if you would like to transition from the visualizations and go back to the statistics and to the raw data, all we need to do is to download this report from Astrolabe. And once we do, we're going to have many different reports going to differential expression analysis and looking at type versus treatment for 15 minutes. We have a spreadsheet with all of the subset and channel combinations. We have fold changes, we have p-values, and so on. One set of columns allows us to see the fold change between each one of the conditions and control media. In this case, we would like to examine the level of PREC1 and compare it between control media and control peanut extract. So all we need to do is to find this column here, and we have the different statistics that we need. Specifically, when comparing the control peanut extract versus control media, we see a fold change of 0.2, while for the patients we see 0.7, and for anti-Ig we see 0.9. For the patients we see 0.8. So I think we can safely argue that these values are all much higher than this one. Plus, we have the p-values down the line in case you would like to be further confident about this. So going back to the platform, the last marker that we saw here is phosphorylation of S6. And once again, we see the same trend as before. We have the controls where the peanut extract does not respond, and we have the patients where the peanut extract response is comparable to anti-Ig. So the last thing I would like to examine here is the element of time. And each of these analyses was done separately for the 15-minute and the 30-minute time points. So I'm actually going to open both of these at separate windows. So what we see here is the 15-minute time point, and this is the heat map that we generated. And switching to the other one, here is the 30-minute time point, and the heat map that was generated for that one. And we can easily compare them and see if there's any differences. So for example, and I'm going to use Zoom to highlight it, if we focus just on the basophils and switch between the two views, then we're not going to see much of a difference. It's about the same. However, let's draw this square around the plasmacytoid dendritic cells. And when we do, we will see that more markers respond for the PDCs in the 30-minute. So this is the 30-minute time point, and we see that PS6, PERK, CD63, and CD61 respond. And if we switch to the 15-minute, 3 of these disappear. So let's focus just on the plasmacytoid dendritic cells and look at PERK1 and PS6. And this is the 15-minute time point. PERK1, we see that there is a bit of a response for the peanut extract. Not as strong as anti-IgE, but it seems to be there. And then PS6, there might be a response, but there is quite a lot of variability here. If we look at the p-value, it's not going to be very high. Then I'm going to switch to 30 minutes comparison and do the same thing. I'm going to pick the plasmatidoid, look at PERC and PS6. Switching to the feature box plots. So PERC, once again, the peanut extract is about a third of the way between the media and anti-IgE. However, looking at PS6, we now actually see a response. So just to recap, this is the figure for the 30-minute time point. And this is the figure for the 15-minute time point. And you can see how no response after 15 minutes, but we do see something after 30 minutes. So to recap, I hope that I managed to demonstrate some of the benefits of using Astrolabe in general, and specifically the new workflow that we have, the new module that we have for differential expression analysis. If you're looking at activation markers, exhaustion markers, phosphomarkers, cytokines, anything related to signaling, this is really the kind of analysis you want to have at your disposal. And doing it with existing tools can be a bit tricky. You need to gate the cells, you need to get the statistics and analyze them. Astrolabe really does all of this for you. And going back to the outline I've shown at the beginning, it's really about providing you with a turnkey solution, a one-stop shop that prepares you for your experiment. And the goal is really to get you from your raw data to meaningful biological insights, help you highlight these trends that are more interesting, the most relevant in your data. So with this, I'm going to remind you that if you have any last questions, please use the Q&A button below. And just as a brief summary, I've shown this figure at the beginning. And I mentioned how as your experiment becomes more complex, the analysis is exponentially more difficult. Based on our experience with labs, with academic cores, with biopharma and biotech companies, Astrolabe is really beneficial in making this curve much more manageable and helping you get from raw data to results in just a couple of hours to a couple of days. So once again, I'm going to thank MetroFlow for allowing this webinar to happen. I'm going to thank you, the listeners. If you have any questions, please use the Q&A button below. I'm going to take a few seconds to let people answer their asked questions, and then I'm going to go one at a time. So the first question is, why not remove dead cells? And that's a great question. So for these specific data sets, they were actually cleaned before they were published. So if you download these FCS files, you will notice that they have no debris, no doublets, and no dead cells. And this is, of course, not the usual turn of events. For your data, Astrolabe automatically removes debris and doublets. It can remove CD45 negative events. And finally, if you do have a live dead channel, such as zombie die, or in the case of CyTOF, cisplatin, you can indicate it during setup and remove it. So for this experiment, they just weren't included in the FCS files. It was cleaned prior to publication. The next question is, how is positivity determined for effector molecules? Are FMOs used? At the moment, Astrolabe does not use FMOs. We're actually looking into that as an option. For the differential expression analysis, the comparison is done based on FMIs. So usually, MFIs, sorry. So usually the purpose of the FMO is to choose where to put the positive, negative boundary for a given marker. I think that in many cases, this distinction is less relevant for activation markers, where the level of the marker is continuous. And yes, there is distinction between positive and negative, but for your experiment, we would really like to quantify different levels of positivity. So the analysis that you see here is using the MFIs for this analysis. One thing that you can set here is a reference feature. This experiment doesn't have any, but if you do have an unstimulated condition, which you would like to use as a baseline, then you can use that. And then, since the unstimulated is not expecting to see any response, that is effectively used to correct the differences between the stimulation conditions themselves. The next question is, are the assignments loaded by the user? So I'm guessing you're referring to the assignments we see here in the cell subset navigator. And these are the different cell subsets that Astrolabe identified. And we can go to the quality control and see the different rules that Astrolabe is utilizing. Astrolabe has a default set of rules, which we call the backbone. And these are tailored to your experiment. I would say about 80% of researchers stick to these default rules, which are provided by the platform. If you would like to customize them, we can work with you to make sure that the cells that we find are indeed those that you expect to find based on your panel. So to answer the question, the assignments can be loaded by the user. By default, they're not. Some researchers prefer it that way. Others want to specify a specific subset. So the next question is, how much does Astrolabe cost? And I just love how these come up every webinar. So Astrolabe is based on an annual license, and the license itself, the fee depends on usage. Essentially, the more FCS files you analyze, the cheaper the price per FCS file becomes. The specifics really depend on your experiment. So if you would like to learn more, and more importantly, if you would like to try a free trial of the platform, please reach out. I'm allowed at astrolabediagnostics.com. I'm happy to answer any questions you have, and I'm happy to give you a trial and discuss our fees as well. So the next question is, how do you deal with batch effects? That's a great question. And I think that whenever you run any kind of experiment, any kind of biological experiment, you should be wary of batch effects, whether it's flow cytometry or anything else. So Astrolabe has several different mechanisms for dealing with batch effects, depending on where you are in the experimental workflow. So first of all of these different cell subsets are classified on a sample by sample basis. In other words, we take each FCS file and we identify the different populations in that FCS file. Conceptually, this is very similar to having a template for your traditional gating and then moving the gates in this template to capture the MFIs on an FCS file by file basis. So that's one way that the platform deals with batch effects. The second way is that when you set up the experiment, one of the columns here can actually be batch. And then all of the statistical analysis I've shown you will also be run between different batches. If you do have any explicit batch effects, then the analysis will highlight it, and you can factor it into your decision-making when analyzing the experiment. And finally, similarly to the question about FMOs, when you set up the differential expression analysis, one of the things you can set is a reference feature. And using the same example as before, if you have an unstimulated sample for every, let's say, patient, then you can use that unstimulated as the baseline. And the batch effects are going to be the same between the unstimulated and the stimulated. So by including this as a reference, you'll actually factor out the batch effect from the statistical analysis. So to summarize, different tools to deal with batch effects, again, depending on where you are in the experiment. The next question is, can you run mouse data? And yes, so both of these examples come from human. We've successfully run human, mouse, non-human primates. There has recently been a new OMIP in Cytometry A for dogs, and we actually analyzed that data as well. We have a video on YouTube that demonstrates it. We've analyzed whole blood, primary blood, bone marrow, lung, spleen, central nervous system, gut, and I'm sure that I missed a few other tissues. We've analyzed tumor infiltrating leukocytes. We've analyzed many different other types of samples. So at this point, we have quite a lot of experience with different organisms and with different tissues. The next question is, what algorithm do you use? So we actually use many different algorithms as part of the platform. And the list is publicly available. Astrolabe is not using any proprietary code or any black boxes. Specifically, you can go to our fact page online, and that fact page is going to have this question, how do I cite Astrolabe? And this includes all the different algorithms that we use. So specifically, to identify the cell subsets, we're using the SOM algorithm, so of organizing maps, along with an algorithm called ECBALA, which we published in Frontiers Immunology last year. And for differential abundance analysis, we use the EDGE-R package. For differential expression analysis, we use the LIMA package. If these look familiar, they were both featured in a paper called DiffCyte, which was published last year. We adopted their methodology for Astrolabe. So many different algorithms in play. And I think the important part is that everything we do, we try to stick to the state of the art. So whenever a new algorithm comes out, we review it and we decide whether it should go into the platform or not. I mentioned that the differential expression analysis module just came out, and we're using this methodology that was published in May of last year. As far as I know, it is currently the best way to analyze this kind of data. So I think we have one last question. So if you want to ask any questions, now is the time. So the last question is, how are the different populations defined? You have a database for automatically defining cell populations. Yes, so if we go back to the assignment level, we have all these different populations, and under the hood, they're found using a combination of clustering and labeling. The labeling is based on rules that come from the literature. So going to this view again, in order to identify the T cells, we're looking at cells which are CD3 positive, lineage negative, and then CD4 versus CD8, and then all these different markers for Tregs, naive, memory cells, and so on. So yes, we do have a database. At this point, it covers about 50 different cell subsets in the immune system. Furthermore, we often customize. So just to give a recent example, we have a researcher who is working on central nervous system in the mouse, and we worked with him to build a specific hierarchy with different kinds of microglia and other cells that you expect to find in the central nervous system. Furthermore, as I mentioned 2 weeks ago, there was a new OMIP for dog samples, and we just customized this to use the labeling hierarchy that they included in their OMIP. Another question, some of my customers have been using Cytobank. How do I switch to Astrolabe? Is it difficult? So I'll start by saying that Cytobank is a great piece of software, and likewise, all the other tools available out there, FlowJo, Omic, I think that all of them are excellent, and they really go to this paradigm I mentioned before, where you have a Swiss Army knife, and you have these different algorithms and the different tools that you can use. I would say that the main differentiator for Astrolabe is that you don't need to make these decisions until much later. In other words, instead of starting from the raw data and cleaning it and running different algorithms and then deciding how to proceed, you can start from the finish line, and you can start from the big level, from the main hypotheses, let's say 5 to 10 major hypotheses, and then backtrack and use the different single cell tools. So I think that switching to Astrolabe should be very easy. Based on experience with our customers, usually they watch 15 to 20 minutes of videos and they're ready to go. The way that I'm interacting with the platform right now is really the way the interface works. So if you want to look at specific subsets, you just click on the cell subset navigator, you click on them here, and then you can scroll down and start making the different comparisons. So for example, once again, there really isn't much of a difference in the different T cell populations across time, across different conditions. So yeah, I would say the transition is very simple, very straightforward. Again, get from your raw data to insights in about a couple of hours and a few mouse clicks. So I'll answer one last question, I think. The cell subsets would have to be customized for each panel that the customer has, or would we have to change our existing panels to mesh with your definition of cell subsets? So to clarify, we never want you to adjust the experiment based on the analysis. As a rule of thumb, updating analysis is easy, updating experiments is difficult. The system that I mentioned is flexible. So we have 50 different subsets in our database, and there's many different ways to reach them. So for example, looking at effector memory T cells, we can reach them using CD3, CD4, CD8, CD27, and CD45RA. Or we can use CCR7 and CD45RO. Or if it's mouse, we can use CD44 and CD62L. Or it's possible that the panel doesn't have CD3, it only has CD4, CD8, and other markers. We can use these to identify the different populations. So to clarify, the system adapts to your panel. If you would like to find specific subsets, we're going to find these subsets. The system is flexible enough to do that. So I'm just going to flash this slide one last time. If you have any questions, please reach out. I'm Elad at astrolabediagnostics.com. And thank you for your time. And thank you once again, MetroFlow, for hosting me today.
Thank you so much, Elad. That was absolutely wonderful. And we also want to thank our attendees for joining us. And please feel free to visit our website, metroflow.org, and visit us on Twitter. It's our Twitter handle is @MetroFlowCyto for any upcoming events. So with that, again, thank you, Elad, and everyone for joining. And please remain safe and happy and healthy. And we'll see you soon.
Thanks, everyone.
All right. Bye-bye.
Bye-bye.